surrounding them, and they have only the center compact core. And in the spiky pellets, you can see there is a compact core with the long appendages. This is a loose mycelium, how the aggregates look. So total 5 cultures produce the aggregates, and the rest 9 cultures were the forming through pellets. So SEM studies were also done, because studies have not been done before on the submerged pelletization. So these were also subjected for the various SEM studies, and the biometeorological parameters were analyzed to give the idea about what exactly this each image says. So the in mechanics, to understand the surfaces, the several parameters has been applied, such as mean density, then motifs, then isotropy. These are the values which are used in the industrial for understanding the surfaces. So in fungal world, they have mostly used the fractal dimensions to understand how much is the complexity in the culture. So we have also tried applying this particular parameters to obtain the result, and found it very relevant, and can be used for separate the different species. So furrows actually indicate how much microporosity is into the pellets, so that will help in a better diffusion of the nutrients and absorbance of nutrients. So based on that, the classification was done. Isotropic indicated the uniformity of the pellet across, so that was also calculated, which was all done by using mountain maps. And the total motive studies was done, which gives a total surface area of the particular pellets. And the fractal studies, which was already done by previously people, and they have also used it for how you see the branching pattern of these species and give the value according to the complexity. So based on this, typological classification was done, and we've given the different categories based on the values, and we found that the different species fall into different categories. And then by correlation, we have seen that the globulus was found to be in the higher range, which was more effective to have a high microporosity, the high total surface area which can take place, the reactions can take place, and complexity is also high, so that to give the good biomass. Hence it was selected as a future for optimization. Whereas the FTI studies were also done to see the different chemical composition of these cell walls across the species, and the analysis was performed, and it was and the binding was assigned to the by comparing it to the different published literature. And it was seen that the amide bands were present in the glucans, which are specific chitin chitosans, have been also identified. So total 50 distinct banding was seen in the Dermatomyces species. So these dendrogram gives you the distances. It was seen there was around 0.06 heterogeneity among these species, and the clustering of these species in the sister clusters were formed at 0.18. So that indicates that how the clustering might have taken place. We estimate that these clusters were not based on the species specific, but based on the metabolite production, or how much is the chemical complexity of the metabolite production, and how close they come together. So based on that, they were clustered. So previously studies have indicated that species from the same genus or the strain, they need not cluster together when you study the various aspects of chemicals. So how the selection was done of the strain, that strain which was highly consistent in production of spherical homogeneous pellet population and highly stable for repeated subculture under submerged condition. Second, the strain which produced a high biomass with low viscosity. And third, pellet lacking long appendages with smooth or finely fibrous lower surface and lacking buoyancy, which helps in the downstreaming processes. It makes it easier. So the dense mycelial packaging with the high microporosity, as indicated by the high furrow density and motif number, surface hyphae that exhibit a fractal dimensions greater than 2.5 and isotropy greater than 60%, as revealed by SEM, were used in selection of strains. So thus we conclude that the Dermatomyces globulus was selected among all the tested Dermatomyces species, which produced the compact, smooth spherical pellets with pellet size ranging from 3 to 12 millimeter, dry biomass of 0.71 gram per liter, cultural broth viscosity of 0.85 cp, furrow density of 11243 centimeter per centimeter square, then total motifs of 1715 and fractal dimension 2.75 with isotropy of 60.5%. So the optimization was done for the pH to see the pH effect on the pellets. It was seen that as the pH increased, there was the reduction in the core size and increase in the appendages. Whereas in the lower pH, it was seen that it was a compact core. This is because the pH influences the cell surfaces, and the aggregates gets loosened up and are released due to the electrostatic repulsions. So in the pH 5 was found to be optimal, producing 1.18 grams per liter, and the second, that was 0.87, 0.98, was not much significance. They were both showing same thing. So the temperature which was selected based on the mount requirement, how the dermatomyces grows. In previous studies, they indicated in the comb and the fungus mounts, that is around 27 to 30 degrees Celsius have been maintained in the mount temperature, whereas so we have selected the range, and it was seen that the pH 5, 1.176 gram per liter was obtained at 28 degrees Celsius, which was ideal for its growth, and at the higher temperature, there was reduction in the biomass. So you can see same in the morphologically that in the lower temperatures and the pH, there was a good production of pellets, which with a good center core, and whereas the higher temperature, it was not that good for its growth. So there was the core reduction size. So the carbon optimization of the globular studies indicated that the maltose was the ideal for its growth, and the sucrose and glucose can be also been used for growth studies, but maltose will lead around the 2.82 grams per liter. And lactose was less because it's not that preferred by the dermatomyces, as it's not found in the fungus comb. There's a lactose source. So the variations across the carbon sources, we could see that the xylose and maltose was inducing the pigment for melanin production. So these carbon sources can be used for dermatomyces species for future for melanin production optimization studies. So previously also there was several authors have been reported that the maltose, sucrose and glucose have been found to be yielding a good mycelial yield, and the solid medial studies on dermatomyces hymi, dermatomyces indicated on solid media that they do prefer glucose, fructose, maltose and sucrose for its growth. So the optimization of nitrogen, the highest yield was seen in the calcium nitrate of 3.3 gram per liter, and the minimum was seen in the ammonium salts. So ammonium salts were not that preferred by the dermatomyces species, and the complex media were also not preferred. This is basically the dermatomyces grow on the substrate which is carbon rich and low in nitrogen, that is fungus doesn't have a high nitrogen content. So maybe that is the reason why this urea, casein, peptones were not preferred by these dermatomyces species. So what happens actually in the ammonium metabolism is that it releases the H plus ions, thus reducing the pH at the medium and inhibits the growth. And it is also seen by the other researchers that observe that those species which have not growing good on the ammonium salts, they might be having an emetophagous activity, and it can be correlated with the presence of rings in the dermatomyces species, which need to be confirmed in future. So various studies have been done on the understanding the calcium role, because calcium plays a major role in fungal cell wall or any other organisms. So the calcium seen that it changes the cell membrane permeability by controlling the internal calcium gradient and activities of some fungal enzyme, which helps in this cell wall expansions. So previous studies also indicated calcium nitrate to be yielding the good amount of biomass, whereas yeast extracts is also reported, but it do have a counter effect because of the rich in protein, amino acids and vitamin contents. So this is the fungal pellet variation on the nitrogen sources. It was seen that nitrates were the good source for producing the black pigmentation in dermatomyces. So with the combination of xylose, maltose and nitrate combination with the nitrogen sources can be used for the melanin production or black pigment production in these dermatomyces. And you can see the poor production of pellets in the nitrogen rich medium. So when the concentration was studied, effect of various concentration of the selected maltose and the carbon source, that is calcium nitrate, it was seen that there is not much significant difference in the biomass yielding, but we got the highest biomass of 3.54 grams per liter at using 0.2% of nitrogen, that is calcium nitrate, and the 3% of carbon, that is maltose. Coming to dermatomyces melanin, This is how the natural fruit body looks like, and it has the melanin amber region. And this is how you can see the initials of fruit bodies of Termitomyces, which are also showing the grey pigmentations and the black spots on the style. So this is mature fruit bodies. It's also observed that the umbo region is black in coloration or grey, annulus also pigmented, and the certain portion of the style and the pseudorhiza appears also black in coloration. This may be due to whence the
Which species is this?
Termitomyces hymen. So during this, what happens is that once the Termitomyces emerges out from the termite mounds, there are a lot of obstacles which are created by the soil in the above ground, and during this there is a lot of abrasions which are crossed on these stipe region, and these injuries are observed to turning to be black in color. So the fruit body melanin was UV visible spectroscopic studies were done. It was seen that there is a linear absorbance from the visible to the UV region, and UV there was the highest amount of absorbance. So the slope studies indicated it has a negative slopes of minus 0.0033. That is because it falls in the range of melanin, the slope. So that also confirmed that it is indeed a melanin. Then the cultural studies was done by using the UV visible. The same absorption pattern was seen in the cultures. And Termitomyces albuminosus was selected as a strain because it produced the consistently black pellets in the submerged conditions, and also the pigment production was consistent in the solid medium. So when you compare the UV visible absorbance, it was seen that Termitomyces fruit body showed a variation because the various parts were examined, and at 223 to 264 nanometers was seen in the fruit body, where solid culture 227 to 273 nanometers was observed. So this was maybe there is a spectral shift was observed because due to the low to high with increase in the molecular sizes of nanoclusters. So in the same studies you will see there is a nano clusters which are seen in these melanin. So this is how the culture looks like in the solid state, and the pellet is forming the liquid. And the previously the Sadiki et al. In 2012 and 2015 reported showed that there is in the culture plate it produces pigment, but they did not identify or as a melanin production. In submerged condition, the biomass studies was done. The biomass indicated that there is 1 gram yields around 14 mg of melanin in the, and the previous studies such as Sun et al. In 2016 and they have worked on the edible mushroom, that is auricularia, and they found that around 2.2 grams per liter of melanin has been produced. So Termitomyces have not been previously described for melanin production. Then these slides indicate that in the first there is the cultures which take up the there is a melanin which is found to be deposited in the cell walls or septate region, and the pellets appears in black to brown. This is due to a reflection of light. The intense black coloration was not seen. So appeared brown in color, and the pellet these plates which are the melanin when subjected and mounted in DPX, it indicated they were very thin thin plates, and the same study indicated that these at the higher magnification they form in the form of nanogranules of the range of 100 to 400 nanometers. Previously the Baltran, Gracia et al. In 2014 reported they have the size of nanogranules of melanin is ranging from the 100 to 300 nanometers. So this also falls in the range, and the size of the melanin particles increases in the hyphal cell wall, known to give a more stability to the hyphal cell. Then the fractal dimensions as studied indicated that it was consistent with the result of Om et al. 2017. The elemental analysis studies was done to indicate what is the composition of the melanin. It was seen that it is a sulfur containing around 12% of the sulfur was reported, with the CN of the 25.6, and auricularia reported to have a 24.4. And these sulfur was qualitatively estimated by using Lysogen's test. So melanin are say basically of major of two types. There is DHN based or it is L-DOPA based. So in the DOPA, what happens is that there is presence of oxygen and thyrosinase. They undergo the crystallization, the addition of sulfur incorporation into the structure, which is leading to the pheomelanin production, which has the sulfur content in it. Whereas the eumelanin, there is no sulfur content, so only nitrogen. So previous studies indicate that is sulfur content in this particular species that 8 to 12% can be present, and because of this benzothiazine and benzothiazole derivatives. And the in 2014 in Lachlan indicated there was around 14% of the sulfur, which is also reported in this species. So our studies indicate that was 12% consistent with this result. So there is a possibility to have such a high sulfur content. So it also, the content of these melanin will change when it comes to the respect to the medium used. So previously the Bull in 1970 has reported they had used the medium of Zepec dogs with the combination of DOPA and catechol, and they showed this carbon, nitrogen and hydrogen percentage, which was similar to our results. Previously in Termitomyces, also both and Acker in 1992 and various other researchers, they have reported several sulfur-containing amino acids. These might be playing a role in the incorporation into their structures and giving rise to pheomelanin. So now also in 2014, Reimer et al. He reported there is a presence of sulfide reductase. So these enzymes helps in the sulfur assimilation. So there is a possibility in Termitomyces to have a sulfur assimilation capacity. So sulfur can be used. And being in these termites which are explored in Goa, it's rich in sulfur bed because it is on the iron sulfide rock. So that is also maybe the reason that it can take up the sulfur content. So our results indicate there is possibly a sulfur metabolism which is present in this Termitomyces, which need to be looked into details. So the UV visible spectral was done in the culture. It was seen it was consistent with the sapiamelanin. Actually, sapiamelanin is not a pheomelanin, it's eumelanin. The peak was obtained as 233 nanometer. The slope was also consistent with the published literature as it fallen in range, and the FTIR studies were done. It indicated the different functional groups, the phenolics, aromatic rings, and it indicated the CS bonding, which was very important to indicate there is indeed a sulfur content. And when compared with the previous literature, we assigned it bandings, the different bands, and we found these 3 bands which were for specific for sulfur. So the EPR studies indicated that it has the higher G value, which in turn confirmed that it is the free radical species which is involved is sulfur, because previously sulfur has been known to have the higher G values, and also DOPA melanins are known to have the higher G values compared to the eumelanins. And coming to the XRD, there was a broad absorbance was seen, and these was indicating that it is amorphous nature or substance, and it forms a non-Braggers it shows a non-Braggers features. Well, that means there are no coherent bonding, which you can see, and then no repeating units. So then the 2D titers, when we compared it to the published literature, we had identified the different reflections, and these are the corresponding reflections. And the NMR studies indeed indicated that there is a sulfur chemical shift that is at 2, and this is nitrogen, oxygen, and this is CH3 alkyl fragments. So when you compare with the others, the same way the assignments was done, and indeed it confirms that it has a sulfur in its structure. So NMR on C13C carbon NMR was done. This is the carboxylic region, the aromatic region, and these are the sulfur-related chemical shifts which were observed when you compare with the others. Similarly, we found at 45 to 40 there was a C carbon assigned to sulfur, which is bound to sulfur. That indicates it is indeed a DOPA melanin. So the MALDI was done to the mass spectrum to see the different fragmentation patterns, what is the different fragments taking place in these melanin, because melanin is very complex structure. So we have seen the highest MZ value was around 778, and there were around 32 different fragments were obtained. So when you compare with the literature, as per Banerjee et al. In 2014, Arun et al. 2015, we see in our present study there was a drop of 2 to 3 units in the molecular weights, which indicates that there is the removal of the hydrogen at the point of junctions to the next to the repeating units. So these are the bands which I said at 32 fragments which were obtained, and these 32 fragments when then compared with the mass calculations, and was seen there was species of hydrogen bound species and with the sodium, which the sodium is the with the addition of the 22 atomic weight mass, sorry. The effect of kojic acid, which is an inhibitor of the dopamine pathway, we had studied the effect. It was seen that the basal media showed the zone area of the melanin around 12, whereas all the cultures, the entire plate was covered, so it was around colony area around 81 centimeter. When you subtract this, you get this particular zone area. So as the concentration increased of the kojic acid at the 100 microgram per ml, it was seen that melanin synthesis was inhibited. Coming to tricyclazole, which is a DHN pathway inhibitor, it was seen that first of all there was a lag in the growth. It took around 20 days to grow, because it may be because tricyclazole is a fungicidal, and also it was prepared with ethanol, so that led to the lag in the culture. But it was seen that the culture grew after 20 days, and it produced the colony was very slow type, and there was the zone area was equal to melanin zone area was equal to the colony. So we see that there is no inhibition of melanin synthesis. So by doing this inhibition studies, we can speculate that there is a possibly that it is a DOPA type of pathway which gives rise to the pheomelanins, as it inhibits the tyrosine to dopaquinone pathway, and this need to further elucidate the pathway in details. So melanin is a three-dimensional polymer for which the number of possible structures is equal to number of type of melanin molecules in nature. And though these spectra which were performed, still it's very difficult to assign a structure to the melanin, natural melanin. So one of my achievement is that I have first time we have screened the 14 cultures of termitomyces, which is large data set, and there have been no information on the submerged pellet conditions of these termitomyces. So we added knowledge in this aspect by using the several scanning electron microscopic and IR based techniques. And we have given the screening technique for the fungal pellets by using the scanning electron microscopic biometrological parameters. And we also reported the first time the melanin in the termitomyces ended by using the several characterization techniques. So these are my published articles. The FTY portions were published in the Journal of Applied Biology and Biotechnology. The purification, that is melanin aspect, was published in the Mycology International Journal on Fungal Biology. The screening of the pellets based on metrological studies were published in Biotechnica and Histochemistry. The Termitomyces bulborizes holomorph detailed studies were published in Kavaka Journal. Then the pellet screening, what techniques we use like viscosity, emulsification, those were published in Journal of Pharmaceutical, Chemical and Biological Science. Then the benefit of the sulfur content or the sulfur rolling in termitomyces in holomorph was published in International Journal of Life Science and Research. Book chapter has been communicated to Springer, and these are the several oral presentations were made and poster papers. We released the poster for the awareness of these particular species to save it, and I would like to acknowledge several people for helping me to do this piece of PhD work. First of all, I want to thank the ST Inspire Fellowship for giving me the financial support, and Dr. Apte for upgradation to SRF. I want to thank my research guide, Dr.Nandkumar, for his endless effort and support for every aspect during the research. I want to thank the past and present Dean of Life Science and Environment. At present, Dr. P.K. Sharma is the Dean, and previously Dr. M.K. Janardhan. Then the past heads and also present heads. The past heads have been several ones at Dr. B.F. Rodriguez. Then now then there was also M.K. Janardhan sir. Then now currently is Mme. Vijaya Ketkar. So I want to thank them and all the faculty members, Dr. Krishnan sir, member Dr. Rupali. I want to thank each one of y'all for supporting me during the entire course of PhD. Especially I want to thank the faculty members and the head of the chemistry, Department of Chemistry, Physics, Biotechnology, even Marine Science, because they helped me in some point or the other of my research. Especially Dr. Nadkarni sir from chemistry, Dr. Tilve, Dr. Srinivasan, for helping me to analyze the various chemical aspects of these melanin. Then in biotechnology, Dr. Savita Ketkar, Then from marine science, Dr. Menon sir, for helping me, encouraging me to write various publications. Special thanks goes to the members of research faculty, Dr. M.K. Janardhan, and my VC nominee, Dr. Sameer Damre, for his valuable suggestion from time to time and thus improving my research work. I want to thank the ex-director of NIO, Dr. Nakwi, who helped me in various aspects, and Dr. Krishna Kumar. And special thanks to the Dr. Singh, Dr. Prabha Devi for providing me FTR at NIO, Dr. Kenneth Rodriguez at Biotechnology Research Institute of Malaysia for helping me in the phytogenical aspects. And still the work is going on this dermatomyces, and we have communicated to him for the various aspects. So I want to thank him also, because these are outcomes which I'm going to publish, and I have to publish. There are a lot of things which I need to publish. Then the
Okay, you have already collected it.
okay.
So,
Okay, then also various like UGC SAFE for providing me chemicals and Sigma and studies, and the other Goa University such as scanning electron facilities, USIC for sputter coating facility, then IIT Bombay and the Central Salt and Marine Chemicals Research Institute, and various other people from the in the Rajiv Gandhi Center for Biotechnology for Matitov, and Dr. Batra Kumar Dabbi, and the also Ganesh Chitgankar for helping me to communicate and get these contacts done and the work to do faster. I want to thank all the MSc past and present students who had helped me, who assisting in the collection of the mushrooms, and the also research scholars from various departments. In some point of the time, they helped me. All the departments actually. I want to thank my parents, grandparents, my brothers and family members, and my mom is present here. Thank you. I want to thank each one of you all for giving your valuable time. Thanks. God bless you all, and Jai Hind.